Hello everyone, welcome back to Equity Tutors. I hope you all had a good Christmas break, and for everyone and anyone who sat exams, I hope they went very well. So today we're going to go back to finishing off a couple of things that we didn't cover last term. Depending on which syllabus and which exam board you are studying under, this is going to be in different modules. So this is going to be just for students who are studying either under Edexcel A or B, WJEC or Eduqas. So if you are AQA or OCR, this doesn't apply to you. And if you are an Edexcel student, if you're an Edexcel B student, this is going to come under the cells module of your AS. But if you are WJEC or Eduqas, this is going to be an A-level topic. But because it's covered by Edexcel in AS, we're going to do it all together today. So what we're going to do is look at reproduction, specifically sexual reproduction in mammals and sexual reproduction in animals. So that's understanding the processes of development of gametes, both in plants and in mammals, and then the process of fertilization. So we're going to start with sexual reproduction in mammals. So I just want to do a quick reminder, quick recap of a lesson that you had last year on cells when you were learning about cell division and specifically meiosis. So some of the basics, and I'll just refresh your memory of that, is that you have two types of cells. You have diploid cells and you have haploid cells. And the difference between the two of these is that diploid cells contain two sets of chromosomes, so the full number of chromosomes, whereas haploid cells only have one set. And the reason this is relevant for reproduction, may be obvious to you, is because when fertilization happens, you want this to occur between two haploid cells. They come together to form a diploid cell. And so gametes are these cells that are haploid and are used as part of sexual reproduction in mammals to enable more genetically diverse offspring compared to asexual reproduction. In humans, gametes are either the egg or the sperm cells, and their development through meiosis involves multiple stages that you need to know about. And the process for an egg or sperm cell is slightly different, and we will go through those individually. So annoyingly, there's a lot of terms that you need to know and learn, and the names of the different types of cells along the path of development. As always, I always recommend that you make a glossary. Unfortunately, this is just one of those things that you just have to memorize. Another tip I always give is to look at as many different diagrams as you can. So I have included this diagram available. If you're watching in video format, you can see the diagram. If not, I always link a document to all the diagrams I refer to below in the bio of the podcast. So I'm going to talk you through the diagrams first for spermatogenesis and then for oogenesis. And as I said, it's good to still look at different types of diagrams because they can vary sometimes, and things like looking at the detail of how many copies of chromosomes are present in the cells at different times can give you clues as to where in the process you are, and different diagrams can have different details that can give you a hint as to where they are. And a very common exam question example would be to show you the picture and say, at what stage in spermatogenesis are we in this picture? So let's start with spermatogenesis. So the primordial germ cells, so diploid cells, which are precursors to the gametes, divide several times by mitosis to form spermatogonia. So this is the top level of this diagram. Spermatogonium if it's one, spermatogonia if it's multiple. Second step, these grow and mature to form primary spermatocytes. That is without any further division, so it's just growth and maturation into a primary spermatocyte. Next, the primary spermatocytes undergo the first meiotic division to form secondary spermatocytes, so these are diploid. And the secondary spermatocytes then undergo a second meiotic division to form spermatids. And these are haploid, but without some of the characteristic of the final spermatozoa that we will get on to discuss shortly. So they do not have the acrosome, the flagellum, etc. But they are haploid. And then finally, we have differentiation. So the spermatids differentiate and grow to form mature spermatozoa. So I'll just go through that quickly again. So we start with spermatogonia. These grow and mature, not divide, into primary spermatocytes. We then have 2 rounds of meiosis. So the first round of meiosis, we get secondary spermatocytes. Then we have a second round of meiosis to get spermatids. These are haploid cells. These all contain the genetic information that each of the spermatozoa cells will include, but they do not have the specialized qualities that the spermatozoa will include. We then get differentiation of the spermatids to form spermatozoa. Okay, so next we have Oogenesis. So this is the word we use to describe the development of the ovum or the eggs. So very similar process to spermatogenesis. So we start with the primordial germ cells, which divide several times by mitosis to form oogonia. So what I mean by that is that it is just a normal cell division in mitosis to form these types of cells. So only one oogonium continues to grow to form a primary oocyte. So unlike the spermatogenesis, which we have lots of sperm, only one oogonium is going into oogenesis to form one ovum at the end. Whereas, as I said, in spermatogenesis, we have multiple. So as I said, only one oogonium through growth and maturation, so no meiosis or mitosis at this stage, will grow and mature to form a primary oocyte. The first round of meiosis then occurs to form a secondary oocyte and a secondary structure that we refer to as the first polar body. And do not worry about these too much. These are just small cells. They bud off the oocyte. They stick to the oocytes, but they do not develop into gametes. But you do need to know that they are there. Then we get a second round of meiosis, so from the secondary oocyte into an ootid. And here, once again, similar to the spermatogenesis process, we get our first haploid cells in this process. But of course, since I just said that we're only going to create one egg or one ovum at the end, this means that when meiosis occurs from the secondary oocyte to the ootid, the other half of the genetic material goes in to form a second polar body. And then finally, we get differentiation from the ootid to form an ovum. And before I go over that again, just a separate note on the timings of when the gametes develop. So the meiotic division for an ootid, to form an ootid, actually starts in utero, but halts at the prophase phase, and only the remaining process and remaining development to form the ovum only occurs in response to fertilization to form the mature ovum. So I'll say that again. The meiotic division starts in utero, but is halted at the prophase phase, and the rest of the development only occurs in response to fertilization. And we're going to go over the process of fertilization in the next section. So I just want to go quickly over the process again. So we have primordial germ cells. They divide several times with mitosis to form oogonium or oogonia. And that is the beginning of the process of how oogenesis starts. We then get growth and maturation of the oogonium into a primary oocytes, and then 2 rounds of meiosis. First round of meiosis, we get a secondary oocyte and a first polar body. Second round of meiosis, we get a ootid and a second polar body. And then finally, 1 round of differentiation which forms the fully formed ovum or egg. So now I want to move on to fertilization, but before we do, I want to discuss the structure of both an ovum and a spermatozoa, so that we can understand when we talk through the fertilization process what each of these structures in the ovum and the spermatozoa is responsible for. Because as ever, as I always talk about, there is a structure-function relationship. Every part of the structure serves a function and is made for their purpose. Starting with the ovum. So the ovum contains the zona pellucida, also known as the jelly coat. So this is a protective coating that the sperm has to penetrate in order for fertilization to occur. And the main purpose of the zona pellucida is to prevent polyspermy. So this is fertilization by more than one sperm. As I'm sure you know, that it is possible to get for more than one spermatozoa to fertilize one egg. That's how we get twins. But it is not super common, and that is because it's difficult to infiltrate the egg because of the zona pellucida. So the ovum also contains a haploid nucleus, so that a full set of chromosomes is restored at fertilization. It contains cortical granules, and these release substances which is responsible for the zona pellucida to become hard, so forming that tough fertilization membrane. There are follicle cells which also form a protective coating around the egg. And of course, as you can see in the diagram, the first polar body is also present there. But these do degenerate and die as the ootid develop. Now let's look at the spermatozoa. So again, the sperm is also structurally functional. And some of those structures that you need to know about are the acrosome. So the acrosome contains a digestive enzyme which will break down the zona pellucida and allow the sperm to penetrate the egg. They contain many mitochondria, which, as you know, is where ATP is generated. So this means that it provides lots of energy for the rotation of the flagellum. So that's the tail, which enables the cells to move and swim to reach the egg. And once again, same as the ovum, it has a haploid nucleus, which allows for the restoration of the full set of chromosomes once the egg becomes fertilized. So now let's talk about fertilization, how those two structures come together, and how these structures help them function within fertilization. So number one, the sperm head contacts the zona pellucida and the acrosome reaction occurs. So remember, the acrosome is the space where there is digestive enzymes which can break down that barrier around the ovum. So the acrosome reaction occurs. Enzymes digest the zona pellucida as the acrosome fuses with the cell membrane of the sperm and then releases those digestive enzymes into the ovum to release those enzymes to digest the membrane of the ovum. The sperm head then fuses with the cell membrane of the egg cell, allowing the sperm to release its nucleus into the ovum. The cortical reaction then occurs, which causes the zona pellucida to harden, and that prevents any further fertilization from occurring. And as I mentioned, that is called polyspermy. So poly being multiple, and then spermy referring to the spermatozoa, so polyspermy. The nuclei within the ovum, so the nucleus of the sperm and the nucleus of the ovum, then fuse together to form a full set of chromosomes, thus forming a diploid zygote. So let's just go over that again. Once again, I recommend that you look at the diagram to help you visualize. So step one, the sperm binds to the zona pellucida, so that's that hardened jelly barrier around the egg. The contents of the acrosome, where there are enzymes, digestive enzymes stored, are released by exocytosis. and the acrosome reaction occurs. So this means that the sperm has released the enzymes into the zona pellucida and digests it, allowing the sperm to burrow through the zona pellucida. When it reaches the membrane of the ovum or the egg, it releases the sperm nucleus. So the haploid nucleus from the sperm is released into the ovum and finally fuses with the egg nucleus to form a full set of chromosomes. Okay, so just to recap what we covered, we looked at the development of both spermatozoa and the ovum. We then looked at the structure of both the human egg and spermatozoa and how that relates to the process of fertilization. And now we're going to talk about sexual reproduction in plants. So it may seem strange, but this is not too different from fertilization in animals. The process is very similar. The only difference is that all the necessary parts are contained within the flower. So one flowering plant will have male and female parts, but it just requires the transfer of the pollen from the male part to the female part of the flower, which is what we're going to discuss. So the male reproductive part produces pollen, and then the ovary is where the female gametes are located. So if we just look to start with at the structure of a flower, I just want you to look at 3 different things. So number 1, the anther. So you may have seen these, for example, in a lily. They're the orange, sort of furry looking structures, and they badly stain your clothes if you've ever touched them. So this is where the pollen is produced. Then you have the stigma. So this is the part in the middle. That is part of the female reproduction organ which receives the pollen. So basically we need to get the pollen from the anther to the central part where the stigma is. And then we have the ovary. So that is at the bottom of the stigma. And that is where the female gametes need to get to. So although it may seem that reproduction in plants is going to be really dissimilar to reproduction in animals, it's actually not. So the anther is going to produce the pollen. They somehow need to get to the stigma, work its way up to the ovary where the female gametes are stored. And the process of forming pollen and forming the ovum are also similar, as they is also a similar process. So once again, diploid mother cells in the anther undergo meiosis, and this forms 4 haploid microspores, is what we refer to it in flowering plants. Then these haploid microspores undergo mitosis to mature into pollen grains. And pollen grains consist of 2 nuclei. So this is where it gets a little bit more complicated. They have a generative nucleus and then a pollen tube nucleus. And there is also a protective coating. So just to go over that again, we start with diploid mother cells, same as we do in animal reproduction. These undergo meiosis in the anther and they form 4 haploid microspores. The haploid microspores undergo mitosis to form mature pollen grains. There's actually fewer steps than there is in spermatogenesis and oogenesis. So we just have diploid microspore mother cells in the anther undergoing meiosis. They form 4 haploid microspores. The microspores then mature into pollen grains. But what is special about these pollen grains is they consist of 2 nuclei, the generative nucleus and the pollen tube nucleus. And we're going to learn about those in a minute when we go over fertilization. Then we have the ovum formation, so a diploid megaspore mother cell note that for the pollen it was a microspore. Now we're talking about megaspore. So a diploid megaspore mother cell in the ovule undergoes mitosis, forming an ootid and 3 polar bodies. And these polar bodies, again, they are degenerate and they are reabsorbed, that we don't need them. The ootid undergoes 3 mitotic divisions to form an embryo sac. Now, this embryonic sac contains a number of different structures which are going to help with the fertilization process. So don't become too stressed out as I list them now, because you will learn to remember them as we go through the process of fertilization. So the embryo sac contains 2 polar nuclei. Together, these are referred to as the endosperm. It contains an egg cell, which forms the zygote, 2 synergids, and these help the generative nucleus reach the egg cell. So the generative nucleus, as I just mentioned, is part of the pollen. And 3 antipodal cells, and these have no established function, but we need to know that they are there, and they all have a big protective coating. Okay, so now let's dive into the process of fertilization, and then we can come back and recap the structures of the egg and the pollen and recap how that relates to the fertilization process. So the pollen grain, which is composed of a pollen tube nucleus and the generative nucleus, adheres to the stigma, where it subsequently germinates. So, as you probably know, how does pollen move? It's usually by the wind. So the wind blows some of the pollen into the middle of the flower, where it sticks to the style and germinates the flower. The pollen tube then grows down the style via secretion of digestive enzymes which digest the surrounding tissues and actually uses those tissues as a source of nutrients. So this is very similar to the acrosome digesting the zona pellucida on the outside of the ovum in order to get access to the egg. So you can see in the diagram here, the pollen tube stick into the style, and the tip of the pollen tube produces enzymes that digest the tissue of the stigma and the style. And that pollen tube nucleus, the one right at the end of the pollen tube, is going to move down into the style. And it is going to move down the style, so it's elongating itself. This is driven by the pollen tube nucleus until it reaches the embryo sac. So the pollen tube nucleus job is now done, and it actually degenerates after the pollen tube has entered the ovule. And at this stage, it passes the job on to the generative nucleus. And the generative nucleus of the pollen divides by mitosis to produce 2 sperm cells which enter the embryo sac. So double fertilization actually occurs. So one of the male gametes, one of the 2 sperm cells that have just been produced, fuses with the female nucleus to form a diploid zygote, and the other male gamete fuses with 2 pollen nuclei to form a triploid endosperm nucleus, which serves as a source of nutrients for the embryo itself. So remember when we were talking about the ovum, we said there was an egg cell which would form the zygote. So one of the fertilizations in this double fertilization process is going to be with the egg cell to form the zygote. And then we also said that the ovum had 2 polar nuclei. And this is what is going to form the triploid endosperm nucleus with the second sperm cell. So let's just go through that again. Let's first recap the important structures from the pollen and the ovum which we discussed at the beginning. So in terms of the pollen, we had the generative nucleus and the pollen tube nucleus. In the ovum, we have an egg cell and 2 polar nuclei. Starting with the pollen, so the pollen sticks to the stigma, and the pollen tube nucleus drives the digestion of the style through the stigma down until it reaches the ovary. So the function of the pollen tube nucleus is to digest the stigma and move the pollen, elongate the pollen until it reaches the ovary. So the pollen tube nucleus then degenerates after the pollen tube enters the ovule. The generative nucleus of the pollen then divides by mitosis to produce 2 sperm cells which enter the embryo sac. And here's where we get double fertilization. So the first of those sperm cells is going to fuse with the female nucleus, so the egg cell, to form a diploid zygote. The other sperm cell is going to fuse with both the 2 polar nuclei to form a triploid endosperm, which then serves as a source of nutrients for the embryo. So those were quite detailed, heavy topics. I think especially the plant fertilization. That was always something that I hated when I was at school. So listen to it a couple of times if you need to. Look at different diagrams, as I said. There's probably also some really good illustrations and animations on YouTube for you to look over. So I am going to wrap the lesson up there today. As ever, we have a bonus lesson out later this week to do some revision on this topic, which I really, really recommend, especially this week as it is very detailed. There's lots of new terms, lots of new concepts to understand. And if not, we will be back next week starting with exchange and transport. So that is the 3rd module of A1 or AS, depending on what you want to call it, and we will be starting that next week, and that is applicable for all exam boards, all syllabuses, so we'll be back all together next week. So thank you so much for listening. Remember, you can access additional content on our Patreon page by searching for Equity Tutors, where we have a second 30-minute lesson every week, plus monthly bonus content. You can also find us on most social media platforms. We will keep you updated on new content, and you can find us there by searching for Equity Tutors UK. Please like, share, subscribe and comment wherever you are listening, and if you're enjoying, please leave a review. Bye!
Bye!